Our next speaker is Aarushi Gandhi. Aarushi is majoring in molecular biology. She's a rising senior. Oh, junior. Yes, see, I'm reading the wrong years. She worked in Patty Mussolino's lab this summer, and Aarushi comes from Bangalore, India.
Right. Hi, my name is Aarushi Gandhi, and I've been working in Patty's lab for about 10 to 11 weeks now. And one of my main projects has been to study the role of the TGF-beta pathway and how it affects the blood-brain barrier disruption that occurs in cerebral ALD. So, cerebral ALD is a devastating and rapidly progressive neurological disorder that occurs due to an X-linked mutation in the ABCD1 gene. So this, as you can see, the brain here at birth looks pretty normal. And then, so the ABCD1 gene affects the peroxisome. So the VLCFAs, the very long chain fatty acids, are unable to enter the peroxisome. So they lead to a lot of accumulation of these VLCFAs. And so because of this accumulation, there is a pre-symptomatic phase where the lesions start to appear in the MRI, and these are asymptomatic. So these kids don't know, or nobody knows that they have ALD at this point. And so after this, for the disease to progress, there is blood-brain barrier disruption that occurs. And this is prevalent in 1 in 40,000 newborns, making it the most common form of cerebral adrenoleukodystrophy. So right after this, these kids that just few years, so like 2 years to like 6 months ago, were playing or going to school, and now they are unable to do any of those things normally, and they start to lose their vision, and they are unable to speak anymore. And so right after that, this is the devastating part of cerebral adrenoleukodystrophy, where in just 6 months to 2 years, they are either dead or they're in a vegetative state. And so my project focuses on this small window that we have necessary so that the blood-brain barrier dysfunction can be stopped or halted, so the progression of the disease from adrenoleukodystrophy doesn't go on to cerebral form. So the blood-brain barrier is basically formed of the endothelial cells, which are in red, and then that's surrounded by an intricate network of pericytes, neurons, and astrocytes. And so the endothelial cells are held together by tight junctions. So this is a more detailed version of this blood-brain barrier, where the red cells correspond to the endothelial cells, and the tight junctions are claudin 5, ZO1, and occludin. So think of this as almost like the customs officer at the airport. They don't allow anybody to enter the blood-brain barrier through into the brain if they don't have the appropriate documents that they need. And so in this condition, this is completely fine. And the increased blood-brain barrier permeability allows the leukocytes to adhere because the claudin 5 tight junction protein is dysregulated. So the monocytes start to stick to them. So the tight junctions become sticky and they become leaky. And right after that, the blood-brain barrier, when it's broken, allows these monocytes and leukocytes to enter through the blood-brain barrier into the brain that causes the inflammation, and it causes the cerebral form of this disease. So to study this, I looked at the TGF-beta pathway, which is what the claudin 5 is expressed through. So TGF-beta, when it activates, goes to the receptor, and SMAD3, which is another transcriptional factor, is phosphorylated, and then it goes into the nucleus of this endothelial cell and then phosphorylates FOXO1, which after phosphorylation leaves the nucleus. So that's an interesting part, because when it leaves the nucleus, it inhibits claudin 5. And so the claudin 5 regulation is downregulated. And so the upregulation of TGF-beta pathways causes the claudin 5 to be downregulated, which means there is no more claudin 5 in the tight junctions for the blood-brain barrier to function properly. So to study this TGF-beta pathway, I created an in vitro cerebral ALD model using HPMECs. So these are human brain microvascular endothelial cells, which are the exact cells that are found in the brain that have these tight junctions. So I can study the proteins in the middle. and see what exactly happens when the TGF-beta pathway is manipulated. So using these HPMECs, I silenced the ABCD1 gene to emulate the pathology that you can see in CALD using silencing RNA and transfecting the cells. And then as control, I had my non-targeting silencing RNA. And then I also had unmanipulated, just normal HPMECs. So after doing that, I treated them with 5 different conditions, and all of them affected the CIS3. So CIS3 affected SMAD3, and then ST for TGF-beta and AS for FOXO1. And I had 2 conditions, 2 different concentrations for FOXO1. And all of these were to see when you inhibit certain parts of this pathway, upstream and downstream, what would happen to the Claudin-5 and the expression and gene expression level of the Claudin-5 and ABCD1. So after doing the treatments, I pelleted them and then prepared them for quantification using Western blots and qPCRs to see the ABCD1 and Claudin-5 gene expression levels. So my results were that the dysregulation of the ABCD1 gene expression is mediated by the TGF-beta pathway. So basically, the inhibition of the TGF-beta and then also inhibition of FOXO1 dysregulated the gene of ABCD1 as a whole. And then when I inhibited the SMAD3 pathway, it increased the ABCD1 and Claudin-5 protein and gene expression levels, which meant that it affected both of them equally, which we think is something to do with the pathway that is influenced. And then finally, I also had TGF-beta, SMAD3, and FOXO1 inhibition, all in different conditions. And we found that TGF-beta inhibition led to the upregulation of Claudin-5 in wild type and ABCD1 silent cells. And then I also found that the SMAD3 inhibition is dependent on the TGF-beta pathway, the canonical one. And then the FOXO1 inhibition is the transcriptional factor, which increases Claudin-5 expression in these ABCD1 silent cells. So in conclusion, I found that the blood-brain barrier destruction is a very critical event in progressing the adrenoleukodystrophy into its rapidly progressive cerebral form. And the loss of ABCD1 function increases the TGF-beta expression, which suggested that there is some sort of codependence between Claudin-5, which is the TGF-beta like expressed protein, and then also ABCD1. And then also the inhibition of SMAD3, which was downstream from the TGF-beta pathway, increased ABCD1 Claudin-5 expression in these cells that I treated. And also the inhibition of TGF-beta leads to upregulation of this Claudin-5 tight junction protein in both the silenced cells that emulated the pathology and also the wild type cells. And finally, the FOXO1 inhibition factor, so the transcriptional factor that inhibited Claudin-5, is a very critical transcriptional candidate in the regulation of this regarding the blood-brain barrier dysfunction. And so all of this data is pretty preliminary, and it's all based on what I've done in the 10 weeks here and also what data the lab has gathered previously. So we need to do a lot more quantification to confidently say that. And then my future directions are to create a blood-brain barrier model using the patient-derived iPSCs, and then so that would be the cells that actually have this disease instead of silencing the gene. And then looking at the effects of ABCD1 gene dose and also the replacement of ABCD1 gene in those cells. And then also confirming the RNA levels to ensure that the silencing has been properly achieved in these cells in the HPMECs that I treated. And then the flow adhesion assay for mapping the leukocytes to be able to see at what point the leukocytes are adhering to these sticky and leaky tight junctions. And by that, we'd be able to tell when the blood-brain barrier is starting to get disrupted, because in kids, you can't see that because it happens during the asymptomatic phase. And then validate the effects of the differentially expressed genes in TGF-beta associated signaling pathways by RNA-seq, and also to see what the constitutive loss of ABCD1 and that would be. And finally, I would determine the functional consequence of this TGF-beta pathway dysregulation and what that affects in the tight junction proteins and also the adhesion molecules that the leukocytes stick to before entering. And finally, I'd like to thank Patty for letting me be in her lab and enthusiastically motivating me to do all the sorts of projects that I wanted to do. Sue for even having this program and like letting me be here and helping me with anything that I needed. And Vijay and Sabya and Jeffrey for being my support system and like helping me learn these different procedures and like Western blots and how to do them properly because I'd never done those before. And Dinara and Colleen for all of their organizational efforts and always keeping everything on track. And of course, my family and my brother for being my personal motivation and inspiration for being here. So yeah, thank you.
Great, thanks, Aarushi. Questions for Aarushi? Excellent job. Thank you so much.